Good morning, everybody, and good morning to everyone on Zoom land. Thanks for being here. My name's Soren. I'm a third-year master's student here at the Douglas, so I'll be moderating the talk today. And I'm really happy to introduce Dr. Valentin Ourry, who's going to be giving our CSA lecture this morning. So Dr. Ourry completed his BSc in biology and MSc in neuroscience at the University of Caen, France. He then completed his PhD in psychology, also at the University of Caen, supervised by Dr. Gaëlle Chételat and Dr. Géraldine Rauchs, where he worked on the effects of lifestyle and sleep and cognitive brain alterations in aging and preclinical Alzheimer's. So the talk today is going to be about modifiable risk factors that can potentially help to halt development of Alzheimer's disease. And so, yeah, we're very happy to welcome Dr. Ourry here to tell us all about resistance and resilience to Alzheimer's disease. So a reminder to everybody, if you have questions, if you're in the room, just say them.
All right, thank you very much for the introduction. I'm very pleased to be here and just want to say thank you for the organization and for the invitation. So thank you very much. All right, so let's start. I will talk about resistance and resilience to Alzheimer's disease here. So we will start with some basic knowledge on Alzheimer's disease and then move on to resistance and resilience, and then how we can measure them. Next, I will show you some in vivo PET imaging evidence suggesting resistance and resilience in preclinical Alzheimer's disease. And finally, I will talk about some underlying biological mechanisms and how we can boost resistance and resilience. So let's start with Alzheimer's disease, which was first discovered by Dr. Alois Alzheimer in 1906 with his first patient, August Dijik. This disease is the most common cause of dementia, around 60 to 80% of cases, and it is characterized by a progressive cognitive decline with memory problems until a loss of autonomy. It is also characterized by two neuropathological lesions, amyloid plaques between neurons, which are composed of A-beta protein, and neurofibrillary tangles inside neurons, which are composed of hyperphosphorylated tau. These lesions and its topography, their progression, have been well characterized by the neuropathologist Echo and Eva Brack in 1991. So amyloid plaque starts to accumulate in the frontal temporal area and extend to the parietal lobe until the rest of the cortex. For neurofibrillary tangles, it starts from the entorhinal and medial temporal lobe to limbic area until the rest of the cortex. And then these two lesions lead to a neuronal and synaptic loss, which finally conduct to cognitive impairment. Since 1952, the neuroimaging era started with the invention of MRI. And with this great tool, we were able to observe gray matter atrophy, but also white matter atrophy with diffusion MRI at the temporal lobe and until the rest of the cortex in Alzheimer's disease profile. And also using fMRI, functional fMRI, we can observe functional disconnection between brain regions, but I will not detail here. And then PET imaging has been invented in 1970 to quantify a target protein in brain. So amyloid tracers have been invented in 2004, and the tau tracer has been invented in 2013. And we were able to match the lesions described by the neuropathologist with a real advantage. The patient is still alive, and we can follow the progression of the disease. All right, so with all this neuropathology and neuroimaging data, a theoretical model for Alzheimer's disease progression has been proposed. So amyloid in red start to accumulate, following by tau in blue there. And then neurodegeneration and cognition match the progression of the clinical stages of the disease. So from a mild cognitive impairment, so MCI, to AD dementia. And what is very interesting here is the preclinical stage, because the physiopathology of the disease starts a decade before the clinical symptoms. So if we can modify the Alzheimer's disease pathology, we should logically delay the clinical symptoms, which is not so simple in reality. Okay, let's move on to the resistance and resilience concepts. I would like to come back many years ago in 1968 with a really old study. So you can see it with the quality of the graph here. So they performed brain autopsy in Alzheimer's disease patients, but also in cognitively normal other adults. And it was really hard to justify to have brain autopsy on normal people at this date. And they observed a typical Alzheimer's disease lesions in 6 subjects here on the graph without cognitive impairment. So it was really surprising. And this study was the first one suggesting a reserve capacity of the brain to tolerate death lesions. Then 20 years after this study, there is another one that confirmed this result with 10 other subjects, so without cognitive impairment. But they proposed an explanation here. So they said that the unexpected finding in the subjects were higher brain weights and greater number of neurons as compared to, and these people may have an incipient Alzheimer's disease, but escaped a loss of large neurons, or alternatively, started with larger brains and more large neurons. And they say they talk about greater reserve here. And from this point, we discovered many genetical and environmentals that can build reserve. So from this previous evidence and further works from Dr. Jakob Stern, he proposed a theoretical reserve model in 2002 with a few updates until 2018. So here you will have the theoretical cognitive trajectory with Alzheimer's disease pathology in individuals with high reserve in red and low reserve in blue. And you can notice that individuals with high reserve are able to maintain their cognition and start to decline later compared to low reserve individuals. So in other words, they are able to tolerate much more Alzheimer's disease pathology. Recently, a few years ago in 2018, Dr. Aurry and Dr. Ourry proposed pathways, a complementary model derived from the reserve model. So the resistance and the resilience pathway. So resistance is defined by avoiding Alzheimer's disease pathology, so where individuals have an absence or lower than expected pathology, while resilience is defined by coping with Alzheimer's disease pathology, so you have pathology in brain. So individuals have higher than expected pathology, so in other words, they can tolerate more pathology in their brain. So which is important here is why and how individuals may resist or being into Alzheimer's disease pathology. And we have identified some contributing factors, such as lifestyle enrichment, vascular risk, sleep, sex, and genetics here. So there are modifiable risk factors and also non-modifiable risk factors. And how about the brain mechanism, but I will detail here. So here is the resistant and resistant frameworks across aging and pathological aging. It is also possible to be resistant and resilient to aging. So from normal aging, you can resist to senescence brain changes here and being resilient to this brain changing regarding the cognition. From Alzheimer's disease perspective, you can resist to the pathology accumulation, amyloid and tau, and being resilient to them regarding the cognition. And then you have contributing factors that can impact resistance and resilience. And then how can we measure resistance and resilience in practice? So for resistance, you will need to have brain features in your models, such as Alzheimer's disease pathology, for example. Then you want to explain pathology by factors, such as modifiable risk factors here. And then depending on your sample, you may have a U-shaped association. So you may have a negative association in preclinical Alzheimer's disease, so at very early stage, meaning that protective factors help to reduce pathology, but you may have a positive association in Alzheimer's disease patients, meaning that protective factors help to tolerate more pathology at later stage. For resilience, which is more complex to assess, you really need cognition in addition to these features. Then you can assess how factors can mitigate the link between pathology and cognition. All right, so in practice, for resistance, you can use linear regression model, basically. For resilience, you may have much more options. So you can either use residuals, so we'll detail here, moderation or interaction model, or also controlling model with different model here. Residuals model have become quite popular over here. As you can see here, there's an increase of publication for residuals here. So basically, the residuals model is you try to explain the error of linear or nonlinear regression model. So between brain features, for example, and cognition here. And a residual is the difference between your observation, or the prompt, and your model. And here, individuals in red have positive values. So cognition is higher than expected. So they are resilient there, and they are non-resilient here. And a recent work from Ellman and colleagues have pointed out the issues of residuals that I recommend you if you would like to use residuals. So the main issue is the fact that residuals could be highly correlated to your cognitive measure. So it may explain cognition instead of resilience. So be careful about that. And I really recommend to read this paper about that. So now I will show you in vivo PET imaging evidence for resistance and resilience in preclinical Alzheimer's disease. So in individuals without cognitive impairment. So I will review some modifiable risk factors such as activity or sleep that could be interesting for non-pharmaceutical interventions for improving resistance and resilience in preclinical Alzheimer's disease. And in the top right of each next slide, we'll have a summary of the factor. So A and T represent resistance to amyloid tau and R resilience. And the white line means that there is evidence with the factor in the literature, and the gray line, no or not enough evidence. So you will have the summary here. All right, so let's start with enriched mental activities, which could be education or cognitive activity. So for resistance, there is a strong evidence in the literature on the link between this factor and amyloid burden, but less evidence for tau. As you can see here, individuals with higher education here or cognitive activity have lower amyloid burden. So here you see that there is a decrease of amyloid burden when you are more educated. For tau, the finding seems quite similar, but less evidence. However, it is done in the multivariate analysis here, so it includes much more factors than just education or cognitive activity. Then for resilience, there's also strong evidence in the literature which enriched mental activities mitigate the effect of Alzheimer's disease pathology and cognition. So individuals with enriched mental activities can maintain their cognition much longer than individuals with low mental activities. So for example, here, as you can see in the middle graph, individuals with higher education in yellow tolerate much more amyloid burden and can maintain their cognition much higher than people with low education in green here. For activity, there is mixed evidence for resistance to amyloid. However, it is pretty clear so this is amyloid, and there is a trend between high and low physical activity regarding amyloid here. But for tau, this is a very interesting study looking at the physical activity and tau in paired twins. So what they did is they calculated the physical activity and tau differences between paired twins. So they controlled genetic aspects in their analysis. And then they correlate these differences and showed that higher physical activity was associated with lower tau burden. So controlling for the genetical aspect. Concerning resilience, there is moderate evidence that physical activity mitigate the effect of Alzheimer's disease pathology and cognition. And here, as you can see, individuals with high physical activity can tolerate much more pathology. So you will have high amyloid burden in blue and a low amyloid burden in red. And here, this is lower physical activity individuals and higher physical activity individuals. And as you can see here, there is an effect of the pathology and cognition here, but when you are physically active, there is no effect of pathology and cognition here. Then moving to sleep, there are many sleep features that have been investigated, such as sleep time, sleep fragmentation, slow wave sleep amount. So for resistance, there is a strong evidence on the link between sleep and amyloid burden, but very limited evidence for tau burden. So here's a very nice review from Andre and colleagues showing these links between within and regions here. And to my knowledge, there is no that has investigated resilience here. For cardiovascular risk factors, such as hypertension, for example, so there is a moderate evidence on the link between cardiovascular risk factors, especially in midlife, and amyloid burden, but it's really a mixed for tau burden. There is no evidence in late life because it could be possible that medication may mask the real blood pressure. And in addition, it is possible that cardiovascular risk may have an independent or synergistic effect. Here, also, it is possible to have vascular disease, which is more related to vascular dementia instead of Alzheimer's disease or pure Alzheimer's disease dementia. In the view of all of this, there is also a mixed evidence for resilience here. Then for neuropsychiatric symptoms, there is no evidence for resistance to amyloid and tau. So here is a nice review from Dennis King and colleagues showing no evidence between anxiety and amyloid and tau PET. in a forest plot, so you will have the effects. There is also a really nice longitudinal study showing no link between depressive symptoms and amyloid and tau burden as well. So it could be explained by the fact that neuropsychiatric symptoms are difficult to measure. It could be more important in midlife, or it could reflect more the consequence than the cause of the disease. And there is no evidence for resilience here. Then move on to personality traits. So even if these factors are not always considered as modifiable, some studies suggest that it could be interesting for future interventions, and it could be modifiable. So anyway, for resistance, there is moderate evidence on the link between personality traits and amyloid burden or tau burden, much more for amyloid than tau, to be honest. And it was summarized in this very nice review with a forest plot with the effect size. And there is no evidence for resilience. All right, so let's move to the next part. So what are the underlying biological mechanisms, and how can we boost resistance and resilience? So how can we resist this pathology? So we can either reduce the pathology production or improve the pathology clearance. So, but first, we need to understand how Alzheimer's disease pathology may be produced. So here you will have the schematic illustration of Alzheimer's disease pathology production in the glutamatergic tripartite synapse. So with two neurons here and here, one astrocyte and one microglia here. So in healthy conditions, so there is a normal neural activity here that leads to release, which is recaptured by astrocytes there. And then there is a control A-beta secretion through the APP cleavage, which is also involved in synaptic plasticity here. And then, so there is a recapture of the A-beta protein by the microglia here, with the help of different transport protein, the ApoE or the clustering here. Then if we move to the pathological conditions, so there is higher neural activity, so very reduced neural efficiency network here that leads to more intense glutamate release. And then there is uncontrolled A-beta secretion, so excessive A-beta secretion with excessive APP cleavage. And at the same time, if the individual is ApoE4 carrier, so which is a risk for Alzheimer's disease, so the transport protein will not work and the A-beta recapture will not function here. And then you will have accumulation of A-beta into A-beta fibril and then amyl deposition in the network. It is still unclear how modifiable risk factors might help resisting to Alzheimer's disease pathology or how it can impact the prediction of Alzheimer's disease pathology. So there is some evidence suggests that individuals with higher physical activity have higher functional networks efficiency and lower amyloid burden compared to individuals with lower physical activity, and exactly the same with enriched mental activities. And there are a lot of animals model with a very controlled environment, showing that if you push the animal with a high intense environment, you will have less amyloid in their brain. And then this evidence can support the better neural efficiency at the end with a decreased amyloid production. In the counterpart, individuals with depression, anxiety, or neuroticism, a trait, may engage in chronic repetitive negative thinking, such as rumination. So it is possibly explain their increased neural activities, especially in the prefrontal and cingulate, where there is amyloid accumulation as well. And then this increased activity might decrease the neural efficiency and increase the beta secretion at the end. But a brief walk in the nature can decrease both rumination and neural activity. So it could be nice to have a walk. Let's then talk to Alzheimer's disease pathology clearance. So the second mechanism here. So again, this is a schematic illustration of Alzheimer's disease protein clearance. So it's just a schematic illustration. As you can imagine, it is more complex than it is. So you will have here two systems for the clearance. So the glymphatic systems, which is an overall system, and very local system, which is the IPAD system, a very early name, which is the intramural periarietal drainage here. So in healthy conditions, so start with the point 1. You will have arterial pulsation here and aquaporin 4, which is water channel protein here that help to make a flow in the brain. So these two systems facilitates the flow here of the liquid in the brain and give a convective flow here in the parenchyma in the brain. And then it helps to move the waste protein, so here the beta and tau protein in the brain, to the venous system through the astrocyte here. Then if we move to the pathological condition here, so there is break of the arterial pulsation perhaps here, and also a break of the aquaporin 4 protein, which are non-polarized here. So there is no convective flow here. And then you will have no clearance of the waste in the brain. So you will have accumulation of these proteins, in particular, A-beta and tau into A-beta fibril and a fibril tangle here. So here, there are a lot of animals models that suggest an impact of modifiable risk factors on the clearance system, but there is no human study here because it's difficult to capture the clearance in human. But fortunately, there are like a few imaging in development using MRI and PET as well. So deep sleep stages have been associated to higher lymphatic system efficiency. So of course, sleep quality is a major contributor of the clearance system here. Also, the physical activity plays several levels here. It could favor deep sleep. It could also reduce inflammation and increase aquaporin 4 expression, the water channel protein that help the flow of the protein in the brain. In the counterpart, poor cardiovascular condition, sedentary lifestyle, stress, or insomnia could alter the clearance system here. So both system, the lymphatic and the hepatic system. And so it will turn into decreased AD pathology clearance. So you will have accumulation of protein in the brain. Yeah, this is what I told earlier on, saying that it is only done on animals model, and there is few sequence that could be very promising to capture the clearance here. Then move on to the last mechanism here, how resilience works. So again, this is the triple synapse here with the two neurons and astrocyte and microglia. So from pathological conditions with Alzheimer's disease pathology and neuroinflammation for microglia and astrocyte as well, individuals with, resilient individuals may have two options. So the first one is to stay in the same network to perform the task. So they will increase their neural activity within the same networks while maintaining a very low neuroinflammation here. But at the end, pathology will accumulate more and more in this network, and it will die at the end. And the second option is to switch the networks within healthy networks without Alzheimer's disease pathology and with normal neural activity and inflammation. The thing here is we don't really know how it works, how it could go to the first option or the second option here. And then about the modifiable risk factors, so regular cognitive and physical activities are known to be beneficial for brain integrity by increasing, for example, neurogenesis, hippocampal volume, or neurotrophic factors. And it will also decrease neuroinflammation, such as macroglia inflammation, oxidative stress, toxicity in brain. And it has been recently shown that these activities, even performed at the age, could increase hippocampal neurogenesis, so the production of, and then it will add for the maintenance and the compensation mechanism that I previously showed you. So this is the summary of the factors that I mentioned here. So we will have the time here, and we will have all the factors here linking to resistance or resilience with the underlying mechanisms here. So some of them, such as physical activity in late life, for example, seems to be very interesting for an intervention because it may be well involved in the physiopathology mechanisms of Alzheimer's disease. So it could be very interesting to look at this factor. And in addition to the factor, the time of the intervention and the targeting population may also matter. So you will investigate or doing intervention on very vulnerable individuals. So without, for example, physical activity to have something significant in this population. I will conclude then by saying that improve the understanding of this model, so the specific modifiable risk factors, working with the pathophysiological mechanisms of Alzheimer's disease will help to design therapeutic interventions. Just a few last words to say that there are much more modifiable factors that I just presented here, and that could be very interesting as well. And in fact, there is 40% of dementia risk that could be removed if we manage the modifiable risk factor. All right, I would like to mention Dr. Sylvia Villeneuve, which is my current PI here, and also Dr. Gaëlle Chételat and her labs. Thank you all for your attention. I will be happy to discuss with you and answer your questions. Thank you very much.
That's great. Thank you so much, Dr. Ourry. Thanks everybody for coming. Thanks for tuning in on Zoom. And thank you to Dr. Ourry for a really interesting presentation. And congratulations again.
Thank you very much.